Thank you very much, Janice, and thank you all for coming to listen. So today I want to talk about a series of experiments that we started about 5 years ago. And the main reason why we started this experiment was we were very aware that when it came to coral reefs, the impacts of climate, or at least the long-term impacts of climate, were not likely to show up until 2050 or maybe 2100. And against that, we were aware that if we want to act to mitigate those impacts of climate, we have to start now, or at least in the next 10 years. And not only do we need to convince scientists that there's a need to change, we also needed to convince industry and the general public that there was a need to change. And for this reason, we decided to take a very empirical approach to the problem, approach where we tried to imitate putting in a reef in a tank. And as you can imagine, that's not very easy, and there's always going to be things that we left out. But we tried to put reefs in tanks and follow them over 2 years approximately, so we could show people in pictorial evidence what reefs might look like as potentially a motivator for changing our approach to CO2 emissions, if that's how the pictures transpired. So the good news in all of this story when we're talking about climate change is it's not a foregone conclusion that we are going to follow the business as usual pathway. Okay? There's still a possibility of following the green emission line here, which could cause much less issues for reefs in the future. Okay? However, if we do follow the business as usual pathway, I left this picture up on the right-hand side here for temperature to show you that not everything stops at 2100. The temperature keeps escalating well past 2100, and we need to remember that because sometimes I feel that we feel the world stops 2100. Okay? The two scenarios we have focused on are the RCP 4.5 scenario and the RCP 8.5 scenario. So where 8.5 is business as usual, and 4.5 is bringing our belts in a little bit with regards to CO2 emissions. One thing I want to point out here at this moment in time is actually 4.5, 2100 is roughly similar to 8.5, along the 8.5 pathway at 2050. And that is important because we're now 2015, that's just 35 years away. Perhaps maybe 1.5 generation times of your average coral. We can't do entire reefs into a tank for an experiment. So we had to choose some place to go to. So we chose to go to Heron Island. Now, last night I was shocked to hear that people think that the Southern Great Barrier Reef is dead. I would like to say it's not dead. There are parts that have been impacted recently badly by storms, but they're growing again. But one of my favorite sites is Harry's Bommy. Okay, and Harry's Bommy, around 5 meters in the water column. You've got large coral cover. You've got large diversity of morphologically distinct corals. So we went there to select our sites. We collected corals, morphologically distinct corals, up to 9 to put into our ecosystems. We selected sediments to go from that site. We selected some vertebrate fish, herbivorous fish, to put into our tanks. We had bioturbators that went in. And when we collected our corals, we also collected some calcareous algae, CCAs, halimeda, a whole diversity. In fact, the whole lab is employed in this collection process because it's a massive collection process when we construct our tanks. The other benefit of Heron Island is thanks to Bronte Tilbrook and Ciro, we had, oops, we're going ahead of time. We have a buoy there that measures every 2 hours what happens to the water temperature and to the PCO2. And for us, that's beneficial because we use that 2-hourly change in temperature and pH to dictate what happens to our control or present-day tanks. rather than the input water that you get at Heron Island that comes from the reef flat where there are no corals growing. Okay, so we could adjust our control to represent the area from which we got the reef. The other nice aspect about Heron Island is there is a vast difference in temperature between summer and winter. And if you're prepared to do a very long-term experiment with a very long-term acclimation period, you can actually introduce the tank, the mesocosms, to future temperatures without actually having to drop them in hot water to get there. It's always a problem. So we start our experiments in April. The natural water temperature starts falling off. For the RCP 8.5 treatment, we just hold the temperature constant, essentially, and we don't have to heat them. It is only when we truly start the experiment in July that the heating begins to apply to them. So we perform them in tanks on the coral deck of Heron Island. That's great. We get lots of tourists come to Heron Island. They love to come and peek in the tanks and see what's happening. So we've got they've got direct access. They can look at any time. They can look in the replicates and see whether they're all performing similarly or dissimilarly. The first series of experiments, we decided to do temperature and PCO2 as combined treatments. We reasoned that they're going to occur together, and if we want to see what the impact is, we should monitor how they interact and the impact that they have together. So at the bottom here, I'm just showing you what happens after 20 months in these conditions where we're following the usual seasonal basis, but with the anomalies of the representative concentration pathways on top of them. As you can see out here, this is our control tank. We were very good at growing corals, okay? And all types of corals, from plate corals to stylophora, to other weedy corals, to brain corals. We put in a whole heap, but they all grew and they all prospered very well. When it came to our RCP 4.5 treatment, this is a bit small, you might not be able to see it very much. We don't have an override of lots of algae. Oops. What you can see is, well, there's a few corals that survived, mainly fungia that made it through the treatment, but most of the others actually died. Okay? And when we came to the red treatment, we basically had very little, or it appears as though we've got very little coral survival, and we've basically got a cyanobacterial overgrowth. So the reefs did not do very well at all. When it comes to looking at the details of what happened to calcification, because we made calcification a focus of this experiment, if you look at the coral-specific calcification rates, perhaps because you can see that a lot of the corals didn't survive in the two future scenarios, it's not surprising we don't see them growing very much either. One noticeable thing is that these two differences are not actually significant. So this is the growth rate throughout the whole of the 20 months of the experiment. Okay, we basically get no growth in under RCP 8.5, but there's not a huge amount of significant difference here. We also determined mini-reef specific calcification, so we used the alkalinity anomaly technique, and we were able to use that technique at 5-week intervals through the full 30 months to construct a picture of what happened over the full course of the experiment. And so we get average daytime rates of calcification versus nighttime rates of calcification. And again, for the daytime rates of calcification, what you should notice is that there's not much difference between the two future treatments, and they're both decreased. What was noticeable, however, at night in the mini-reefs, okay, so it's not just corals in our mini-reefs that are calcifying, there's a whole lot of other things, but we see a huge decrease in calcification as we went up the types of scenarios that you might experience. When we looked at the microbial biomass, okay, we focused on sediment microbial biomass. What we found is that with treatment, we get an increase in that microbial biomass. We're going through the compositional data at this moment. At a brief, fast look, there doesn't seem to be much change in the composition, although we think some of the rare microbes might be increasing relative to the others. No, I can't go forward. Okay. So I think those responses are alarming, okay? I think those responses are alarming for the reason that 4.5 is not very far away, and actually our reefs under 4.5 are hardly growing or not growing at all. Another reason why it's alarming is when we looked at which are the corals that are winning out of the subset that we put into the tanks, it tends to be solitary polyp corals, okay? Aptasia, okay? We didn't put Aptasia in our tanks. It might have come in with our tanks, but under RCP 8.5, we had thousands of them generated in the tanks, okay? And they were everywhere, which is why I'm showing you this one stuck to the bottom of the Holothuria, okay, a sea cucumber. So they did very well. The other corals that did well were also the single polyp corals, and what we found is that through the first summer they bleached, through the winter they recovered, through the next summer they started to bleach again. Okay, so they survived, they did well enough, but even those corals, and this is just the fungia-specific buoyant weight changes that we observed, they decreased their calcification rates. Okay, so the corals that survived stopped calcifying. Okay, our experiment, we had no storm activity in it. Okay, so, you know, one thing is clear, that to recover from a storm, as we're seeing on Heron Island at the moment with the storm activity that's happened on the north side, you need very fast growth rates, okay? And this is just absent from these future treatments. So, some people are not prepared to mitigate CO2 emissions and are suggesting that perhaps we should engineer our way out of the problem, okay, and perhaps limit either the temperature aspect or the acidification aspect through geoengineering. Before we go down that track, we really need to understand whether reducing one of those impacts is going to have a major effect on the outcome. Personally, I think this is a bad idea because putting mirrors in space to repeat is likely to have other impacts on reefs, such as a reduction in light, which I do not think is somewhere we want to go. Nonetheless, we do want to know what the individual impacts of temperature and acidification are. So when we finished the first experiment, we then started the second experiment. And in this experiment, we have isolated the effects of temperature from the effects of acidification in 2 of our treatments to see what's going to happen. At this moment of time, it's an ongoing experiment. We're not due to finish this till March 2016, but I thought I'd give you a sneak preview of what we're seeing. Okay, so this is our present day tank, and it just reinforces the fact that we are well able to grow corals in a tank, okay? So I've just picked out the key moments in time. I picked out December because we've done this with RCP 8.5 temperature treatments. We should be seeing bleaching in this by December. April, because it's after the first summer, and September, because it's the last pictures we took just before I came here. If you look at what happens when we just increase PCO2, actually the tanks look really good, okay? One thing that is noticeable is Halimeda seems to absolutely love it, okay, and is doing really well in our tanks. Potentially, there's a slight impact on some of the corals, okay, but there's variability, and every time we do this, we see some corals die, and in other tanks, others survive. So I want to run this longer to see what the longer-term impact is going to be. When we look at the temperature, the treatments when we've just added temperature, one thing that I find is noticeable, at least in the early parts of this experiment, is the absence of algae in the tanks, okay? It's actually fairly algal-free, okay? But what you do see is we see our fungia. Interestingly, it's not bleached. You can see the crop is starting to go in this tank. By April, they're definitely bleached. Most of the other corals, apart from this Lobophilia, are dead. And then in September, we've got recovery that is occurring. If we look under the dual impact, when we heat and we acidify, okay, what we can see is we have bleaching that is definitely in place by December in this treatment. We don't have bare space. We have a lot more algae already present in this treatment. But as you can see here, the bleaching, we actually had earlier recovery in this tank than we did in the other treatment. But when we get to the end here, we've got our halimeda present, but we've got a lot of bare space, a lot of dead coral. Going with this, the coral-specific growth rates are principally temperature affected at this moment in time. But I think that is principally driven by the fact that we've got high mortality in those treatments. Temperature leads to mortality. Mortality leads to reduced rates of calcification. If we look at mini-reef-specific calcification, and I've just focused on December and January here, between December and January, we have a 1.2 increase in baseline temperature across all of it. What's noticeable in December, the only driver for decreased calcification is nighttime decreases in calcification driven by PCO2. However, in January, when the temperature is just that little bit hotter, we see that temperature takes over as the major driver for decreases in calcification. So in summary, the types of corals that appear to be able to survive under RCP 8.5 and 4.5 under both temperature and PCO2 do not promote reef calcification, okay? Specifically, they will not promote the recovery of reef from episodic storm events. This is not good news because current policies, under them, we are heading towards the 4.5 response by 2050. Less than a generation, well, 1.5 generations of your typical coral lifespan, okay? And furthermore, the initial results just suggest that temperature is the main offender because temperature leads to mortality under these conditions, but that PCO2 nonetheless has an insidious effect, especially on nighttime calcification rates. Where I really want to go is next is one, I understand why single polyp corals seem to do so much better than when they're in the colonial form. And secondly, understand what is driving this nighttime dissolution that we observe. Whether is it coral-based, is it something in the sediments, is it microbial-based? Thank you.